you're going to be watching is on differentiation and development. So the idea here is, how do cells decide what they're going to become, right? How do your skin cells know they're going to become skin cells? How do your liver cells know they're going to become liver cells? Because every cell in our body has the same genetic information, right? Every single cell has the same amount of DNA. So how are we turning on and off certain genes, and when? So to go through this video, we need to have a little background knowledge, so some terminology. So the first thing is differentiation. So differentiation is just going to be cells changing from one type of cell into another. So they're going from a less specialized state into a more specialized state. And this happens because of induction. So they're being induced somehow, those cells. So induction is just the influencing of one cell. So one cell influences another's growth and development. So induction in turn then is going to influence that differentiation. So development is just the organism on a whole, how it's going through that growth and differentiation. So during embryonic development, a fertilized egg gives rise to many different types of cells, and that's because of this differentiation process where we're turning on and off genes. So when we turn on and off those genes, it's going to affect that gene expression. Alrighty, so here is just a basic overview of embryology. So morphogenesis is just the organism developing kind of its shape and spatial arrangement, and its distribution of the cell through embryonic development. So if we look from when they first become fertilized, become that zygote, so from the zygote stage to about the 4-cell stage, at this point we are still totipotent, so we're undifferentiated. After that, when we become our morula, and then go ahead and become the blastocyst, we become a pluripotent stem cell, so which can become any body cell, and eventually we specialize even further and become multipotent stem cells, which some of them become multipotent stem cells. Alrighty, so here, all I want you to do here is kind of note the difference. So we have an egg cell, and you see various sperm cells. So in this video, we're really not going to get too into how only one sperm cell will enter the cell and fuse, and then in turn, right, we only want one sperm cell to fuse because if you're getting multiple, it's too much genetic information. So if you're interested, I highly recommend going through your text, examining those ways and what occurs as far as chemically to prevent that fusion of multiple sperm cells, because we only want one sperm cell in that egg. But what we are going to focus on are the two types of chemicals. So once the sperm cell enters the egg and we have formed that zygote, what are the types of chemicals that will then influence our differentiation and set up the embryo? So the ones we're going to be focusing on are cytoplasmic determinants and inductive determinants. All right, so if we take a look here, when you check out the unfertilized egg, right, you're seeing these two chemicals. So the brown cytoplasmic determinants and the green cytoplasmic determinants. So at this point in the unfertilized egg, they're still pretty random. Once that sperm fuses with the egg and we have fertilization happening, you see more of a distinct one side being green, the other side being brown. And then obviously after fertilization we have mitosis happening, so we're going to split those cells. And now once we have our two cells, you could really see each of those cells already starting that differentiation process because both now have pretty different cytoplasmic determinants. So these brown chemicals are going to signal to turn on certain genes, whereas in this cell where they don't have the brown genes, they're not going to turn on that gene. They're going to turn on a different set of genes. Alrighty, now we move on to the induction determinants. So induction, again, is just influencing, so one cell influencing another cell through some sort of chemical signal. All right, so we see here at the bottom our early embryo, 32 cells, is going to be releasing this chemical signal. All Right, so we have to remember our kind of inducers and receptors here, how that affects the signal transduction pathway, which then goes ahead and turns on certain genes. So depending on how much of this chemical signal you're receiving, so the closer you are to that signal, the more of it you're going to receive. My cells at the top probably are not going to receive as much of that chemical signal, so we have a gradient going on here. So that gradient is going to affect what type of cell you turn into and how you're going to differentiate. And there's actually a lot of research being done about embryonic development and how they're not quite sure what turns on the cilia on the outside of these cells, but they think that cilia helps basically send up the chemical. So this release of the chemical, the cilia turns on and creates this current in the womb. Of those cells receives that certain chemical is going to determine what genes they're able to turn on and off. Alright, so here's kind of a cool example of an experiment that was done where they wanted to try and actually induce the genes themselves. So it was an attempt at experimental, I guess, scientific induction. So in this fish, right, they're called blind K-fish, so they do not produce the functional eye. And what they were able to do was take a fish's eye, basically try to implant it or put it near where the eye should be. And what they saw was in this embryonic eye, it was releasing chemical signals, so doing induction. And where they had placed the eye, those cells actually made an attempt at trying to form a functional eye. So we can see it didn't do this successfully, but it is proof that, you know, that eye must have been releasing some sort of chemicals, and then at the gap junctions it was taking in those signals and at least making some sort of attempt to then differentiate and create that functional eye. Alrighty, so determination is going to commit a cell to its final state. It happens because genes are turned on and off, which causes differentiation. So determination actually comes before differentiation. So you can think of differentiation is when we are producing our transcription factors. Right, so we need to create those certain proteins. Those proteins or transcription factors then are going to help turn on certain genes, right? They help turn them on and off, and then in turn do differentiation. Alrighty, so we see in this first embryonic, so this is just an example for you, an embryonic precursor cell. You can kind of tell it's going to end up being a muscle cell based off of the shape. So at this point, right, our master regulatory gene is turned off. So at this point, we are not totipotent anymore, but we're pluripotent. But the master regulatory gene is off, so not differentiated and not determined just yet. Once we are a myoblast, now we've turned on that master regulatory gene. That master regulatory gene is going to be, now we're determined. Right, so we're creating, we have that transcription factor, but the genes are still off. Right, so we're determined, but we are not yet differentiated. Then, once the transcription factor is created, we can now turn on all these other genes and differentiate to become our full muscle cell. Right, so there's going to be layers of these master regulatory genes, and what's really cool is that these are trackable through evolution. So you and I and all other vertebrates have these master regulatory genes, and it allows us to kind of track evolution and see the changes in embryonic development that are occurring. Alrighty, so now we move on to pattern formation, which is just the development of spatial organization of tissues and organs. So think like, where's your head going to end up? Where's your butt going to end up? Alrighty, so pattern formation begins with the establishment of these major axes, so the anterior, posterior, again, head and butt. The positional information just helps determine where this pattern formation is going to end up. Alrighty, so we're going to do an example with Drosophila flies. So this isn't necessarily going to be true for every single organism, but it is a really nice example. In Drosophila, it's very simple, and it shows us where the head and butt, anterior, posterior, are going to end up. Alrighty, so we see here our adult fly. We have the head at the anterior position, abdomen at the posterior position. Alrighty, and just giving you a brief overview of kind of what happens in their development. So we have an unfertilized egg here, so you have the egg cell. You have these group of nurse cells occurring towards the left. As the egg gets larger, still unfertilized, your nurse cells begin to deplete, and then fertilization occurs. Right, we're going to have mitosis happening, and then now we see this segmented body, the embryo forming more through all those divisions, and we see a pattern start occurring. Right, you have the head at the anterior, butt at the posterior, and then larval stage, again, staying with that same pattern formation all the way to adulthood. Alrighty, so I just want you to keep this kind of image in mind as we move along. Alrighty, so one thing I want us to focus on are these maternal effect genes. So they're going to encode for cytoplasmic determinants. They are present in that initial unfertilized egg, and then will later help set up the head and butt, and we'll talk about how they do this. But those maternal effect genes, they also are called egg polarity because they set up, obviously, the opposite sides of the body, right, where the head's going to be, where the butt's going to be. Alrighty, so the most common maternal effect gene is called a bicoid. So it's a bicoid protein. It's a morphogen, which determines where the head is going to be. Alrighty, so this is just a chemical. It helps set up that physical structure. So the bicoid is a cytoplasmic determinant, sets up where the head will be. If that gene is mutated, your head will not get created. You're going to create two butts, in fact. So let's see. Alright, so here at this top picture, the wild type, you have a normal functioning bicoid protein that was produced. So the head was created. Posterior, you have the tail. In our mutant larvae, you see something was mutated with the bicoid protein and that maternal effect gene. So we created two tails. Not so good of a scenario here. Alrighty, so let's take a closer look at how this is all occurring. Alright, so here you can see a concentration gradient of the bicoid mRNA. Right, during fertilization, again, we see a large concentration here. And on the other end, you don't have a concentration at all. So posterior end, we don't have any of that bicoid protein being created, so you're not going to create a head. At the anterior, you do have that bicoid protein, so the head will be created in that location. Alrighty, so how does this all happen? So we see the bicoid protein in the early embryo, so that came from the bicoid mRNA. And if we look at the bicoid mRNA, you could see those small depleted nurse cells. So this is prior to fertilization. Right, so we can kind of infer, argue that those nurse cells were inducing, right, the bicoid mRNA. The nurse cells were releasing a chemical signal and influencing how these cells would differentiate or how they were going to create the protein. Alrighty, so this gets started the process of cytoplasmic determinant, and then the gradient, right, is going to help set up that entire organism or the entire fly on a whole. Right, and again, we call them maternal effect genes because these are coming from only the mother. Right, this is an unfertilized egg, these nurse cells that are inducing the egg. Right, so three important points about the bicoid is that one, it's identified a specific protein required for pattern formation. It's also helped us really understand the mother's role in embryonic development, right? Those maternal effect genes, how they begin that inducing process to help differentiate the cell later on. And it's also demonstrated that a gradient is going to affect polarity, right? Where the head's going to be, where the butt's going to be. Okay, so going back to the master regulatory genes, right? We notice the pattern in the adult fly through to the embryo, and then we could see it on the chromosome here. On the chromosome are the master regulatory genes, right? So we also call them homeotic genes or Hox genes. And we can see them in other organisms as well. So here you have just another example of these Hox genes where we can actually map out on the chromosome itself that pattern formation, right, where it's going to occur. So you can see where the head's going to end up, segmented body, the posterior, right? And again, same thing goes for the adult mouse. So we've already set up that body orientation early on in these Hox genes, right? So they're already there, they just need to be turned on. So if you have any questions, we're going to talk about this more in class. Please make sure you're referring to your textbook. And if you have any questions, make sure to ask them in class. Thanks so much.